Costa conference. I was supposed to give a general talk that included some references to the future of tropical biology, right? So what better than going into the TARDIS, right? Excellent. Okay, so we know that we are losing species at an unprecedented rate, right? And we think that by 2050, between 15 and 35% of all species on Earth might be risk of extinction. And we know the main generators, right? We know that it is habitat loss, species invasion, overhunting, and climate change. And here we have the typical representation of a tropical mountain and its different life zones, right? We have tropical rainforests, we have pre-montane forests, we have lower montane forests, we have mountain forests, and we know that with an increase of between 5 to 6 degrees in the next century is what's going to happen, right? We're going to have a shift of ecosystems up here. In the lowlands, we're going to have these biotic attrition, new temperatures, new weather regimes. But one thing for sure that will happen on top of those mountains is that those high elevation ecosystems will shrink or they might even disappear, right? From my lab, from my research here in Costa Rica, we know a few things about how organisms will respond to climate change. We know, for example, here we have the Barba elevation gradient and the Taramanga elevation gradient. So the north of Costa Rica, the south of Costa Rica, we have the two mountains, the elevations, and all of those lines represent the elevation distribution of beetle species. And we know that beetle species that are present at higher elevations are less prone to survive under projected climate change, so they are less tolerant to high temperatures, right? And also, we're working on plant-insect interactions and what is going to happen with changing either host plants of different temperatures, right? So here I have three representations of one new theoretical framework I'm working on that we call demographic networks, right? So we all are used to seeing these bipartite networks where we describe who is what, right? This beetle species, is this plant here, and so on, right? And people use these models to try to understand extinctions and co-extinctions. For example, what happens if I remove this host plant here? We're going to lose whatever species are connected to them if they have nothing to eat, right? That's the assumption of all the current models that we have for co-extinction from plants to insects, right? However, one thing that we're doing in Costa Rica at La Selva Biological Station is we are actually measuring fitness of those interactions, right? Actually, we're measuring how long these beetles can survive in a given host plant, how many times can they reproduce over time, and if you connect fecundities and longevities, you can measure one thing that is super important, fitness itself, right? So what we have here is, this is the first demographic network, a network of interactions where we have plants, insects, who is what, but this measurement that you see here is the actual fitness, right? Black, populations are stable. Blue, population decline. Red, population growth. So look at this. If you look at the diets of these insects, this beetle species that was supposed to eat 3 host plants, actually, this host plant here, actually you have population decline in this plant, right? And if you have an extinction simulation in this network, and you remove this host plant here, it is super counterintuitive, but actually extinction of this host plant will be beneficial for the overall population growth rate, because actually you have 2 plants where you can actually obtain population growth, right? And another thing that we're doing in Costa Rica at La Selva Biological Station is that we are bringing insects that are present at different elevations, right? For example, this beetle species is one species of beetle that is present in the lowlands at La Selva, populations that are in montane forests on top of this mountain, right? Then we raise all these insects in different incubators at different temperatures, and we measure fitness. And look what we find, right? For the lowland elevation species, you have all the different temperatures here. They are able to survive only between 15 and 20 Celsius degrees. 15 Celsius degrees and lower temperatures, they won't get fitness. Same thing for higher temperatures, right? Look at the high elevation species, population, right? You have the opposite. If you look at those results there, you can see that those populations of the same beetle species, they are locally adapted to temperatures, right? So the idea of this project is that we want to combine demographic networks from the perspective of the diets, from the perspective of their temperatures, to actually estimate what is happening with the fitness of these insects with climate change, right? Okay, but if we really want to understand what is going to happen from the perspective of physiology and demography with these interactions, what we need to do is actually to understand what is happening all over the world, right? So how can we do that? Imagine that you had a device that you can actually use to travel instantaneously to any place on Earth, and that you can actually measure different fitnesses, the fitness of these different organisms in different diets, different temperatures, and so on, right? That would be amazing, right? And actually, when I started my job at University of Connecticut, and I had this meeting with the architect designing my lab, he asked me, okay, so what do you want for your lab? And I said, I want this device that you open the door, you get in, and you can be anywhere on Earth. And they were like, what are you talking about? How can we instantaneously be in all these elevations and latitudes? And so I told the architect, this is what I want. I want the TARDIS, right? So I don't know if you guys are sci-fi fans, you know what is the TARDIS, right? So Doctor Who has this ship that is called the TARDIS, the time and relative dimension in space ship, and then you can get into the TARDIS and you can be anywhere in the galaxy, right? And the cool thing is that the technology that we have now allows us to actually build TARDI, right? For example, you can build an incubator, right, that can replicate exactly the same temperature of La Selva and humidity right here, right? Or anywhere on Earth. You can have the weather of the Himalayas in your lab, right? The only issue is that to actually build a TARDIS is super expensive. So I want to introduce you to a project that I have with some colleagues in Mexico that we call the $500 Laboratory of Interactions and Climate Change, right? And the objective of this project is to design accurate and inexpensive equipment to estimate thermal limits and demographic rates of ectotherms at different elevations and latitudes. And the idea is that you bring a bunch of equipment to these workshops, and students come with their ideas and different projects. And then this is a workshop that we did in Mexico, and after that we just build stuff, right? And we build the devices, and then we have all the different designs for different questions. So here I'm going to show you really quick some of the devices that came out from these workshops. So here we have this super inexpensive device that can measure the critical thermal maximum, the highest temperature at which organisms can faint. And actually, it was a design from an OTS course, and part of the data produced here was published in a journal called PNAS. And here we have this other CT mean device that is a miniature freezer that you can bring to the field, and you can actually cool down your organisms, right? It is too small for a beer, but it is big enough for a beetle or something like that, right? Super tiny, it's like this big, right? Another examples. So this is a design of a hack of a fridge that you can buy anywhere. And then you connect this heating element, and then you can build this temperature controller, and then actually you can have an incubator for less than $300, right? Or here we have a prototype of a device that my postdoc just built for an experiment in La Selva. So here we have a plant, one of the plants we work with in La Selva, and we have all these communities of insects that they live in these raw leaves, right? So what we built was a device that is portable that you can bring to the field, and then you can just put it around. This is a young leaf of the plant, right, where all these insects are, and you can change the temperature. So what we have here is the actual temperature logged outside. This is the ambient temperature, and the two green lines are two different treatments of climate change in the field. So you can use these to increase temperatures in these raw leaves, but also, for example, in flowers, and see what happens when you actually heat flowers, what happens with pollination, or if you have, I don't know, little water pools in a bromeliad, you can actually heat them using this same device, right? Okay, so these are some examples of the different tools for the TARDIS, right? So here we have our team at La Selva Biological Station building the TARDIS. We have this amazing workshop where they can build anything for you, right? So we have here Canuto building the TARDIS, Aaron cutting stuff, maybe putting together one of the incubators. And actually, we contacted a bunch of colleagues with this idea, and they are super excited, right? So we have one TARDIS that is at the University of Connecticut that is finished, and we use it in the spring, so we are ready for the experiments to start happening this spring. We have a TARDIS in Mexico, where all these students are doing all these different experiments. The TARDIS at La Selva in Costa Rica, that if you guys go to La Selva, please let me know, and I can show you our TARDIS. So you can go all over these different elevational gradients. And recently, a team of researchers in STRI, the Smithsonian Tropical Research Institute. Doctor Who, are you here? Okay. This is super cool. Are you okay, Mahmoud? Mahmoud is totally fine. Yeah, he's unbreakable. All right, so in Panama, my colleagues this summer will build their own TARDIS, right? So what we're gonna do is, we're gonna have all these experiments happening at the same time with different organisms, right? But we're gonna be trying to answer similar questions using all these different devices. Okay, so I want to welcome you to the Project TARDIS, the Thermal and Altitude Research in Dynamic Invertebrate Systems. Okay, so I want to just highlight a few projects from the TARDIS at La Selva. One of the main processes generating extinctions is one that we never think about because it is the most pervasive. So what we have here is a quote from the tropical biologist and second professor in the OTS fundamentals course, Dan Jansen, that says, what escapes the eye, however, is a much more insidious kind of extinction, the extinction of ecological interactions. So here we have examples of organisms that depend on other organisms, right? We have a host plant, we have its insects, the insect herbivores, we have its pollinators, we have their seed dispersers. So what is gonna happen with all these different interactions if the host plant disappears, right? So at the beginning of our project, we started working with the one that I think that is the interaction that is more closely associated with the plant, more specialized, that is herbivory, right? Interaction between plants and their associated herbivores. So in a project on the process of coextinction, what we're trying to do is to estimate the rates of extinction and coextinction amongst complex trophic networks, right? So this project is developed along the La Selva Barba elevational gradient. That is this amazing place. This is the highest elevational gradient of continuous forest in Central America. And along the elevational gradient are the study organisms that are kind of like the core system in our laboratory. So these are plants from the order Zingiberales. So these are the banana-like plants, the prayer plants, the gingers, right? And there is a group of insect herbivores that radiated in the neotropics. These are called the Cephaloleia beetles. That is right here, right? And Cephaloleia beetles are also called the roll-leaf beetles because you have these young leaves that are rolled in these host plants. And if you unfurl any of these young leaves in the neotropics, this is what you will see, right? Communities of insect herbivores living in those rolled leaves, all right? By the way, the beetles don't roll the leaf. The leaf is rolled because it is young, right? Okay, but when the leaf matures and opens, then beetles have to fly and they have to look for another host plant that has a young leaf, right? And they get back into the rolled leaf. So to model coextinctions of insect herbivores in elevational gradients, we need the following information. First, the elevational distributions of plants and insect herbivores, plant-herbivore associations, and some measurement of the strength of the interaction between plants and herbivores, right? For example, in this case, we know that this beetle species fits 50% on this host plant, 30% on this host plant, 20% on this other host plant, right? So here is a representation of our study site. So at the base, 3 meters, is La Selva Biological Station, where we have our laboratory. And in blue are the different shelters along this elevational gradient, right? And what we did at the beginning of this project was to go all over this elevational gradient, and then we identified the elevational distributions of all the plants and all the associated insect herbivores, right? And after doing this is a tropical elevational gradient, right? So bam, diversity happens, right? Imagine that. So if you want to understand coextinction, you need to understand which beetle fits on each host plant, how their diets change with the elevational gradient, but also you need to know the proportion attributed to each one of these host plants to the overall dryad breadth for every single beetle species, right? So that's a complex issue, right? So, of course, as a good scientist, right, what I did was to shelter in fantasy, thinking, what can we do to actually identify all these interactions, right? So this is actually the cover for one interview for, so Mary Kirksbreaker, she's a journalist from the New York Times, and she wrote this piece about our project, right? So I don't know if you guys remember, or maybe the older generation will not remember, right? But in the original series of Star Trek, you have Mr. Spock and all the people traveling in the Enterprise, and they are scientists, right? What they are doing is basically what resource people and students do in the OTS courses, only that they don't go to field stations, they go to planets, right? And Mr. Spock has this amazing device that was called the tricorder. I don't know if you guys heard of this, right? So the description of Maggie, of the device, is the tricorder is a device that looks like the bastard love child of a Polaroid camera and a 70s era portable cassette deck, right? It is portable. It has this super swanky leather strap that you can actually use it to walk around with your tricorder. It is portable, right? Thing is, the tricorder is not cool for how it looks, but for what it can do, right? Imagine that Mr. Spock could go to any of these planets, get a piece of any organism on Earth, put it in there, and actually know the species, and then it's connected to some other mega library, and you know the natural history and so on, right? It would be awesome to do that, right? So the solution for this, right? Everybody heard of this through Jan Jansen, because Costa Rica is the center of generation of this idea, right? DNA barcodes. So what I did was to develop this technique where you can actually identify the host plants of these insects by using DNA. So the way it works is we put together the full library of DNA barcodes for the plants along the mountain. Then after that, we developed this technique where you can extract DNA from the gut contents of the beetles, and then you can match the diets, right? And then at the end, what you get is something like that, right? All the different interactions between insects and plants using these DNA barcodes, right? So what we have after this analysis is you have each box represents a plant or a beetle species, who is what, but also because you have molecular data. I'm not going to talk about that now, but you have also the phylogeny, the evolutionary history of all these interactions. And the full data set for this mountain looks like something like this, right? You have all the interactions of all the beetles with all the plants along the elevational gradient, and also how these diets change as you switch different elevations, right? So do co-extinction rates of insect herbivores increase with elevation. So I'm going to show you just a super short couple of figures, a summary of the results of this, right? So here we have, this is one of the measurements that we use to estimate risk of extinction of insect herbivores as host plants disappear. And these are just the random models, right? So here we are in the lowlands, and you have and then you have, this is the trajectory of extinction of each beetle species at each elevation, right? And these are the confidence intervals that you are present or absent. Let's go up the mountain. Co-extinction happens faster, right? Even faster. Super fast. You remove one host plant, everybody will potentially be at risk of extinction. What is going to happen here? You have only one host plant for beetle species. So what is going to happen if this plant disappears, right? Basically, and one of those insect herbivores there that is more sensitive to temperature is Cephaloleia cressi, that I named after John Kress. So, all right, but the plot thickens, because when these beetles fly out of those raw leaves, they are not flying alone. These beetles are covered by phoretic mites. So these are mites that they have to hitchhike on these beetles to be able to move from plant to plant. So you have this universe of interactions. You have mites that are symbiotic. They feed on a fungus, Rechia, that grows on the elytra of the beetles and might kill the beetles if the mites don't feed on the fungus. You have filter feeders, so they just detach and they start feeding on like the water in the leaf. You have mites that are predators of nematodes. You have mites that are predators of other mites. So it is this universe of interactions, right? So for this project, we are trying to estimate the rate of extinction from plants to insect herbivores to phoretic mites, right? And of course, right, you have a big issue here, that is that you need to now to know the species of mites hitchhiking on each one of these beetles, right? A huge diversity of mites. Basically, for one beetle species, you can have 12 mite species that are specialized on that particular beetle, right? So you have this immense diversity. So this is the data set that we have so far
for the different elevations, right? And what we learned so far from the different analysis is that in the lowlands, mites seem to be more specialized to insect herbivores. However, when you go up the mountains, mites are more specialized on the plant species. Well, these are data from a colleague from Occidental College. So she's working on the bacteria in the gut contents, right? That we can actually connect to these models of coextinction. And this is more or less an illustration of different interactions we're working on, right? So we have the beetles feeding on the plants. We have the mites on the beetles. We have bacteria on the gut. But also we need to think now in the pollinators. So now we have these different projects that are focusing on interaction between the pollinators and the plants, right? So in this case, you can have here a hummingbird. And I don't know if you know this, but hummingbirds visit flowers, and there is this particular group of mites that they live in flowers of hummingbird pollinated plants. And these mites feed on nectar, and the only way they can move from one flower to another flower is by hitchhiking on the beaks of the hummingbirds to move from one plant to another, right? So here I have, this is a video from one of my students, so I want you to pay attention to this interaction. This is from one of my students at the University of Connecticut, at La Selva. Look at the beak. You see the little dot coming down? There's another visit. And look at this, another mite going down, right? This is super fast, right? So for this project, what Laura Bizzarri is doing is that she has these high-speed cameras at La Selva, and she already recorded all the interaction between plants and hummingbirds, right? She collected tens of thousands of mites from the plants and also from the beaks of the hummingbirds, and now we're going to be able to know which mite species is living in each of these different plants, and which mites are hitchhiking on the different hummingbirds, right? So that's super, super cool, right? So here we have, let me see, it's coming back. Look at this, going down there, you see it? Now it's going to do another visit here, and you will see that the mite goes down, right? That's super, super, super cool, right? There you go. Okay, so things that are happening right now in Laura's project. Now she's able to figure out, identify which mite species are hitchhiking on all hummingbird species of La Selva. She's identifying all the mites that are inside the flowers, that are hummingbird pollinated. But this is new, right? So this is super cool. So this is eDNA. So we are almost ready to have really good identifications of the pollinator when visiting the flower. So imagine that. You have DNA that you collect from a flower, you rinse the corolla, and you can actually get DNA from the insects or the birds that were visiting these plants. So that's totally Star Trek, right? I don't think Mr. Spock was able to do that with the tricorder. Maybe yes. Okay, so this is a little bit of publicity. Students, you can join the TARDIS. You can be the next Dr. Kien. So we have 2 positions to join my lab. All right? So talk to me. Okay, so I'm going to do a very short trip to the past and future of tropical biology. Kyle, I did my homework. Danielle, this is not as great as yours, but there you go, right? So we're going to take a little ride through tropical biology. So did you know that we need to thank Fidel Castro for creating OTS? The story is, Fidel Castro, during the revolution, told Harvard that they had to leave the island, that they had a botanical garden field station, and they had to leave. So Edward O. Wilson, professor at Harvard, and Jay Savage got together and said, what are we going to do? We're not going to have a tropical biology station anymore. What can we do, right? So what they did was, so Jay was a friend of Dr. Rafael Lucas Rodriguez, right? And we need to thank Don Rafael and many other Tico scientists for funding OTS. All right, did you know this? Jay Savage paid for La Selva with a bag full of gold. You guys heard this? So imagine that. So when Leslie Holdridge wanted to sell La Selva to OTS, Jay Savage said, okay, so how much should I pay for it? And this is true. You can ask Julie Denslow. You actually interviewed Jay, and he told you the full story. And then Holdridge told him, yeah, I can sell it to you, but you need to bring some gold ingots in a bag, and then I will sell it to you. So he had to fly from Miami, I think, and he had to fly in an airplane with a bag full of gold to pay for La Selva. Okay, and this picture, this is a really good description, right? A Santa Monica beach baby. Okay, you know this? Carl Sagan paid for part of the land to connect La Selva with Brillo Carrillo National Park. Isn't that amazing? So that shows actually the impact of OTS all over the world, right? So actually, there is a paper in Science, a letter to Science written by Carl Sagan and other scientists, where they asked for money, and they were willing to pay the same amount of money that people donated to this cause to buy the piece of land to connect Braulio Carrillo National Park with La Selva. And in the letter, Carl Sagan said that he was really worried that you had this amazing tropical rainforest that will become a fragment if this didn't happen. And we bought the piece of land that connects Braulio Carrillo to La Selva that I cross every time that I go to look for my beetles and my plants. Okay, and this is me, right? So when I was an undergrad in Colombia, I came to the OTS course, first time that I left my country. And then after that, Giselle Castro and Barbara Luis made sure that I had a job before I went to grad school. And eventually, through OTS, I ended up meeting my advisor, Carol Horvitz, and also my postdoc advisor, John Kress, right? So impact for everybody. For example, I was able to meet Dave Janus, professor at University of Miami, and first director of La Selva. His job was to give towels to people when they arrived to La Selva, right? This is great. The TARDIS just took us to this amazing moment, the Stone Bridge inauguration, right? Imagine this bridge, right? How many times we cross this bridge just to discover the tropics? Okay, and the big connection with the TARDIS, right? The OTS courses. Here we have a picture of the first OTS course by Dan Jansen, Connection to the Past. And this is the OTS course that just happened, the Latino course. There's the connection with the future, right? So I'm going to close my talk with a last question. What is the TARDIS, right? It is OTS that is connecting all of us to the tropics, and we all are here because of that. Thank you very much.